Hi everyone, I am Dr. C.S. Kulkarni, MD, and I am going to teach medical biochemistry. Before I start the topic, please like, share, and if you are new to the channel, please subscribe it so that the next video will be independent to you. In the last session, we ended up with regulation of glucogenesis. In today's session, we will start with functions of glycogens, in which we will see that glycogen is the storage form of carbohydrates. Then we have glycogenolysis, in which we will see that glycogen phosphorylase removes glucose as glucose 1 phosphate from glycogen. Then we have debranching enzyme as alpha 1,6 glucosidase. Then we have branching enzyme as amylo alpha 1,4 alpha 1,6 transglucosidase. And lastly, we have regulation of glycogen metabolism. Good morning, everyone. Let us start the topic. Glycogen is the storage form of carbohydrates in the human body. The major sites of storage are liver and muscle. The major function of liver glycogen is to provide glucose during fasting. The glycogen content of liver is 10 gram per 100 gram per tissue, whereas in skeletal muscle it is 1 to 2 gram per 100 gram of tissues. But the total quantity of muscle glycogen is more than liver glycogen because of the larger muscle mass. When blood glucose levels fall, liver glycogen is broken down and helps to maintain blood glucose level. After taking food, blood glucose tends to rise, which causes glycogen deposition in liver. After about 5 hours of taking food, the blood glucose tends to fall, but glycogen is raised to glucose so that the energy needs are met. After about 18 hours of fasting, most of the liver glycogen is depleted. When depot fats are hydrolyzed and energy requirement is met by fatty acid oxidation. The function of muscle glycogen is to act as reserve fuel for muscle contraction. All the enzymes related to glycogen metabolism are cytoplasmic. Coming to the next point, glycogenolysis. Glycogen phosphorylase. It removes glucose as glucose 1 phosphate from glycogen, that is phosphorylases. It contains pyridoxal phosphate, that is PLP, as a prosthetic group. The alpha 1,4 linkages in the glycogen are cleaved. It removes glucose units one at a time. Enzyme sequentially hydrolyzes alpha 1,4 glycosidic linkages till it reaches the glucose residue 3 to 4 glucose units away from the branching point. It cannot attack the 1,6 linkage at the branching point. If glycogen phosphorylase alone acts on glycogen molecule, the final product is an highly branched molecule and it is called limit dextrin. Coming to the next point, regulation of muscle glycogen phosphorylase. Skeletal muscle glycogen is degraded only when the demand for ATP is high. The regulation of glycogenolysis in skeletal muscle is by epinephrine and calcium released from sarcoplasmic reticulum. Glucagon has no effect on muscle glycogenesis. AMP formed by degradation of ATP during muscle contraction is an allostatic activator of phosphorylase. The calcium binds calmodulin and the complex activates phosphorylase kinase. Coming to the next point, debranching by bifunctional enzymes. Then a block of 3 glucose residues, that is trisaccharide unit, are transferred from branching point to another branch. This enzyme is alpha 1,4 glucotransferase. Now the branching point is free. The alpha 1,6 glucosidase, that is the debranching enzyme, can hydrolyze the remaining glucosyl units held in alpha 1,6 linkages at the branching point. Here we can see glycogen molecule is acted upon by phosphorylase, then by glucan transferase, and finally by T branching enzyme. This glucose residue is reached as free glucose. At this stage, the ratio of glucose is 1-phosphate to free glucose is about 8:1. The transferate and alpha 1,6 glucosides will together convert the branch point to a linear one. With the removal of the branching point, the phosphorylase enzyme can proceed with its action. Coming to the next point, phosphoglucomutase. Phosphorylase contains phosphorylase reaction produces glucose 1-phosphate, while debranching enzyme releases glucose. The glucose 1-phosphate is converted into glucose 6-phosphate by phosphoglucomutase. In this diagram, we can see glucose 1-phosphate is converted into glucose 6-phosphate by the enzyme phosphoglucomutase. Coming to the next point, glucose 6-phosphatase in liver. Next, hepatic 6-phosphatase is hydrolyzes glucose 6-phosphate to glucose. This free glucose is released to the blood stream. On the other hand, muscle lacks glucose 6-phosphatase. The important point is muscle will not release glucose to bloodstream because muscle tissue does not contain glucose 6-phosphatase. Instead, in muscle, glucose 6-phosphate undergoes glycolysis to produce ATP for muscle contraction. Coming to the energetics. In muscle, the energy yield from one glucose residue derived from glycogen is 3 ATP molecules, because no ATP is required for initial phosphorylation of glucose. If glycolysis starts from free glucose, then only 2 ATPs are produced. Coming to the next point, glycogen synthesis. The glycogen synthesis occurs by a pathway distinctly different from the reversal of glycogen breakdown, which would prevent the operation of utile cycles. The steps are: first, activation of glucose. UDP glucose is formed from glucose 1-phosphate and UTP, that is uridine triphosphate, by the enzyme UDP glucose pyrophosphorylase. Glucose 1-phosphate plus ADP gives UDP glucose plus PPI in the presence of enzyme UDP glucose pyrophosphorylase. The glucose moiety from the UDP glucose is transferred to a glycogen primer, that is glycogenin molecule. The primer is essential to accept the glucosyl units. The primer is made up of protein carbohydrates complex. It is a dimeric protein having 2 identical monomers. An oligosaccharide chain of 7 glucose units is added to each monomer. Coming to the second step, glycogen synthase. Glycogen primer containing n number of glucose units plus UDP glucose gives glycogen plus n plus 1 number of glucose units as UDP in the presence of glycogen synthase. In the next step, the activated glucose units are sequentially added by the enzyme glycogen synthase. The glucose unit added to the non-reducing, that is outer end of the glycogen primer, is formed of alpha 1,4 glycosidic linkages and UDP is liberated. Here you can see that glycogen is converted into extended form by the enzyme glycogen synthase with the UDP glucose converted to UDP. Then this glycogen with the extended form is acted upon by the branching enzyme and alpha 1 6, and at alpha 1 6 this glycogen glucose units bind and this cycle is repeated. The next point is branching enzyme. The glycogen synthase can be added glucose units only in alpha 1 4 linkage. And a branching enzyme is needed to create alpha 1 6 linkage. When the chain is lengthened to 11 to 12 glucose units as residues, the branching enzyme will transfer a block of 6 to 8 glucose residues from this chain to another site on the growing molecule. The enzyme amino alpha 1 4 alpha 1 6 transglucosidase, that is a branching enzyme, forms the alpha 1 6 linkage. To this newly created branch, further units can be added as alpha 1 4 linkages by glycogen synthase. Next point is regulation of glycogen metabolism. The synthetic and degradative pathways are reciprocally regulated by to prevent futile cycles. The phosphorylated form of glycogen phosphorylase is active, but glycogen synthase becomes inactive on phosphorylation. The covalently modified phosphorylase is active even without AMP. Active, that is dephosphorylated glycogen synthase, is responsive to the action of glucose 6-phosphate. Covalent modification modulates the effect of allostatic regulators. The hormonal control of covalent modification and allostatic regulation are interrelated. These hormones act through the second messenger cyclic AMP. The covalent modification of glycogen phosphorylase and synthase is by cyclic AMP mediated cascade. Specific protein kinases bring about phosphorylation and protein phosphatases cause dephosphorylation. Here in this diagram we can see epinephrine and glucagon in liver and epinephrine in muscle activate adenylyl cyclase, which increases cyclic AMP. This cyclic AMP stimulates glycogen synthase and glycogen phosphorylase, which leads to increase in glucose 6-phosphate and glucose in muscle and liver respectively. In this diagram, we can see glycogen synthesis, which is active or dephosphorylated, converts glucose to glucose 6-phosphate and it gets inhibited. In this diagram, you can see epinephrine or glucagon attaches the receptor on the cell membrane and inactivates adenylyl cyclase, which leads to formation of cyclic AMP. This cyclic AMP increases active protein kinase. This active protein kinase then stimulates conversion of active phosphorylase to active phosphorylase, which leads to the active glycogen phosphorylase. Then this glycogen phosphorylase activates conversion of glycogen to glycogen N-1 plus glucose 1 phosphate. Here ends our session. Kindly like, share and subscribe the channel. Thank you.